introducing our speaker, Wesley Knapp, who is the mountains ecologist and botanist for the North Carolina Natural Heritage Program in Asheville, North Carolina. His talk is Tales from the Crypt, the Extinct Plants of North America, with a focus on New England extinction events. I'm going to stop sharing my screen here to prepare for Wes to be presenting, but I'll give a little bit more about his background now. He holds a Bachelor of Science degree in Environmental Sciences from Catawba College in Salisbury, North Carolina. He also earned a Master of Science in Plant Science with a focus on plant systematics from Delaware State University. He worked for many years with the Maryland Department of Natural Resources in the Wildlife and Heritage Service until taking his current position with North Carolina Natural Heritage Program in 2016. In his work, he explores and inventories some of the most sort of botanically fascinating natural areas in North Carolina and by extension, Eastern United States. His work identifies conservation targets for prioritizing sites for acquisition, conservation, and management. Wes is an expert in numerous plant groups, including the extinct plants of North America that he'll be speaking about this evening. He's written major treatments of the rush family, the Juncaceae, and he's a regional reviewer for the Flora of North America project. He's described several new plant species, run studies of natural communities, and is a lead author of a forthcoming annotated checklist of the vascular plants of Maryland, and is a founding member of the Digital Atlas of the Maryland Flora. He has a long list of scientific publications, including a high-profile lead authored paper on the extinct plants of North America. It was published last fall in Conservation Biology, and is what we're very excited to hear about this evening. Additionally, he teaches plant identification workshops, and his current research interests focus on endemism, identifying extinction events, preventing extinction events, and describing undescribed species. Wes is also a native New Englander. He was born in Lawrence, Massachusetts, and then raised in Claremont, New Hampshire, and has many family across the region. So it's great to have him back to speak to the New England Botanical Club this evening. So without further ado, I will hand it over to Wes.
Wonderful. Well, welcome everybody, all you out there in internet land, friends and family from across the region, I guess. It's weird to be back in a Zoom meeting. Haven't been to New England in probably a year now because of the pandemic, but maybe someday I can get back up there and give a presentation in person, because it's neat to come back to a region where I kind of cut my teeth and develop my love of the natural world. And one fun little update, you'll notice on the screen here, when I submitted my CV for this talk earlier this week, this wasn't part of it, but I am now with the UNC, University of North Carolina, Chapel Hill logo here. I am now in their PhD program in ecology. So hopefully you'll be getting a lot of good work out of the weekly lab about extinct plants and documenting extinction. And this is going to be a two-part talk tonight. We're going to talk about the extinct plants in North America. I'm going to focus on all the New England events. And then I'm going to talk about the biodiversity and wonder in all of Western North Carolina, just to wet your whistle a little bit. Maybe you'll have me back someday. As Jesse mentioned, this talk I will be giving tonight is largely based on a paper that was published in Conservation Biology, first online in August, and then finally physically published in February, just last month. This is an open access article for anybody who wants to get it. And this probably took 5 years or so of dedicated work. And this paper is not the Wes Knapp show at all. This is an amazing group of co-authors to lead that really humbled me. So like Anne Francis, who's Nature Serves botanist, she was my number 2 on this. I can't tell you how many 10 p.m. phone calls we had trying to figure this out. Reed Noss is just one of the biggest names in conservation down in Florida. And like Rob Noxie is probably the local guy for y'all up there. He's at New York Botanical Garden, rewriting Gleason and Cronquist's manual. But this authorship was amazing to work with, and it really is a bunch of wonderful people from across the country. But the background to this project, am I frozen? There we go. So the background of this project starts when I had my first professional gig as a botanist. Right out of undergrad, I moved from Salisbury, North Carolina, and I had got a full-time job with the Maryland Natural Heritage Program. And I was all of a sudden tasked to go out and inventory Maryland and find rare plant species. And that was really overwhelming. Like I felt like I was a decent field botanist at that point in my career, but I just wasn't ready for the task. So I spent a lot of time studying the rare plants of Maryland. And that list is a real dry list of just scientific names, common names, and then rankings. And we'll talk about those rankings later. But the plant that captured my imagination, even from those early days of study, was this one here that I've highlighted. Not all is micranthemum, micranthemum because it was the only plant in the entire flora that had this rank of GH, or which means globally historic. And then there's an X next to it, which means extinct. So that captured my imagination. Like, whoa, there's extinct plants in Maryland. I need to learn everything I can about this thing, and maybe we can rediscover it. And this is the plant in question. Not all is micranthemum, micranthemoides. It really isn't much to look at. You have to be a botanist to love it. It is what I playfully call the opposite of charismatic megafauna. Because you think about, lions and tigers and bears capture the imaginations. In the plant world, we have orchids and carnivorous plants. This is about as extreme as you can get for a vascular plant. It's very depauperate. It's a small thing. It isn't really beautiful, but I guess beauty's in the eye of the beholder. It was found in a very difficult to survey habitat, these intertidal mud flats. So it occupied the sweet spot between being too freshwater or being too saline, and it would only occur in mud flats that drew down to have a very nice exposed muddy bank. And the last time this plant was ever collected was the 13th of September, 1941 in Tidewater, Virginia. And it historically occurred from New York all the way to Virginia. And it apparently wasn't even rare. Fernald and Long collected, this is actually a photograph of the last known collection. There are 3 duplicate specimens of this. This is a specimen at the Academy of Natural Sciences in Philadelphia, but y'all have one at Harvard and there's another one at VPI. And the story goes that Fernald and Long were out just documenting more occurrences of this plant for the upcoming Gray's Manual of Botany. And that was the last time anyone ever saw it. So I felt like that was a fascinating story. Why don't I know this as a botanist? And I talked to other botanists in the region. What plants are extinct in your area? And if I gave this talk live in person, I like to ask the audience, how many people know about Franklinia altamaha? And I usually see a lot of hands. The Franklinia tree is without a doubt the most famous extinct plant in the North American flora. It's taught in most botany books for students. Lots of common mass media has been devoted to this plant because it has a fascinating story. So this plant was collected a single time was collected a few times as a herbarium specimen. But thankfully, Tom Bartram, who was traveling the Southeast, who wrote a book called Bartram's Travels, encountered this plant, and it's in his book right here on my bookshelf. He actually says, basically, I'm summarizing, I traveled down the road in Georgia. I saw this tree that I'm going to name after Benjamin Franklin, and that was one of the last times it was ever seen by man. So thankfully, he introduced this into the horticultural trade. It was from the banks of the Altamaha River, named in honor of Benjamin Franklin, and now you can buy this online. It's at numerous gardens. But the story to me, what I found fascinating is that even professional botanists didn't know what plants were extinct in their own regions. So I did a literature review and I found out this was a big knowledge gap. Like you can find books written about lost animals, lots and lots written about birds as you'd almost expect. And there was even a really nice paper about extinction rates for freshwater fauna, but there was a big data gap when it came to the extinct plants. And that's what caused that authorship that I talked about to get kind of big. Is first question was, has this work been done? And I missed it somewhere. And then it was, well, how do you handle a project of this scale? So the goals of the project were rather simple, but they took a lot of time to get through. We wanted to first compile a comprehensive list of all the plant taxa, which is species, subspecies, and varieties, believed to be extinct in the continental United States and Canada. We wanted to calculate some extinction rates, because as I'm sure we've all seen in the newspaper, they're predicting horrible things for the future. So we wanted to set a baseline for the Anthropocene. And if you don't know that word Anthropocene, that is believed to be the current geological era that we are currently in. So we wanted to calculate an extinction rate as of today to test against an extinction rate as we move forward in time. I wanted to draw attention to these extinctions because I wanted to help drive future conservation efforts. Because I'm not an academic scientist, I'm like a more pragmatic field ecologist, I'm a conservationist. So what can we learn from these untold stories of extinction? And is there a way that we can learn things and then prevent future extinction events? Now, this does get kind of into the weeds, and I'm not going to spend a lot of time on this because you can spend a lot of time and a lot of ink has been spilled in papers and books about presumed extinct versus globally historic. Now, when I say extinct in this talk, it means presumed extinct because extinction is a hypothesis. We can only disprove this hypothesis. And as botanists, hope springs eternal. We hope up the next river or over the next hill, we're going to find one of these extinct plants. But at this point, the data does not suggest they're still in the wild anywhere. So each one of these species I'll talk about today has been searched for by numerous people at numerous locations where they historically occurred. We have no confirmed sightings for more than 25 years unless the only place for them has been destroyed. And for this project, we're going to look at extinctions since European settlement. And furthermore, we're not talking about extirpations because there are some plants that are extirpated from the United States, meaning that they're no longer found in the continental United States or Canada, but they're still found in Mexico or a Caribbean island. So we're truly looking at extinction events. Now, the study area is an artificial study area. I want to be clear about that. But it largely parallels the Flora of North America project, which I'm sure we're all familiar with. It's a 28-volume account of all of the plants of North America, north of Mexico. Now, their project, Flora of North America, includes Greenland. Our paper did not, but I've subsequently dug into the extinct plants of Greenland, and there are apparently none known. I will point out that this study that I'm going to talk about today does not talk about bryophytes, but there is apparently one extinct moss from Canada. I won't even attempt to pronounce that name because I don't know how to say it. And we excluded Central America from the study because, frankly, it's still relatively poorly known. Lots of new species are being described, and we don't want to come to a conclusion about extinction rates that isn't well-founded. And the study does exclude areas like Hawaii and Puerto Rico because then you have issues of high endemism and island biogeography because Hawaii is itself an extinction hotspot. I believe there's 98 extinct plants just known from Hawaii. So if we include Hawaii in this data set, it really kind of doesn't tell the full story about the continental landmass of North America. The methods are rather rudimentary. It took a lot of time in the library. Like I spent days at the Smithsonian, for instance, going through floras, lots of work in the primary literature to see what was known about these plants. And I also polled botanists from across the US and Canada. As a member of the Natural Heritage Program Network, I get to meet some excellent botanists, like Bob from Massachusetts invited me here tonight. It was that contact, that connection, and that network that allowed this tale of extinction to really be told, because we have a phenomenal data set from across the United States and Canada. Now, along with the literature review and talking to local botanists, we also did a data query looking at GH, globally historic, and GX, globally extinct plants. But those tags, GH and GX, are tags we don't use a lot. So I work with the North Carolina Natural Heritage Program, and our data fills to the national level and the international level through NatureServe, this conservation organization that hopefully you've all learned about. So when I say a plant is globally rare, it comes with some authority that it's globally rare because that data has been vetted. And we use a nice extinction matrix here with 5 being the score of most common and secure, 1s being critically imperiled, the most rare. And then we have these tags H and X, which hopefully we don't use because that means they're gone. You'll see SH and SX a lot more frequently because they've been extirpated at the state or local level, whereas GX and GH are much more uncommon to use because they're globally extinct. But when you put a data query like this, you end up with a lot of different names, and not all names are created equal. As botanists, I'm sure we understand that. So at one point, we had 150 names that could be globally extinct or historic, but we didn't know what that meant. So because you'll end up with plants like Franklinia, that Franklinia tree I talked about at the beginning. Every botanist agrees Franklinia is a distinct entity. But then you end up with things like Rubus fryi. Now that is either an extinct plant from West Virginia that was only known from one place, or it's not real. And those are 2 very extremely different options. Because if you rediscover this plant, it would become an immediate global conservation priority if you recognize it as real. So we had to come up with a way to deal with all of these names in a standardized fashion. And we didn't want to just use one flora because you're getting one person's opinion if you do that. So we developed the Index of Taxonomic Uncertainty, the ITU. So that is the scientific consent of a name. And it's a calculation only by using novel taxonomic treatments. So I would check, for instance, Gleason and Conquest, who are the Flora of the Northeast. and Arthur Haynes treatment and Weekly's treatment to see how they treated these names in the floras. We did not use databases, so even NatureServe, we didn't accept those names from NatureServe. So we wanted to go to people who spent their careers trying to separate these plants based on characters to see if they agreed or disagreed that it was distinct. And I had this silly little cartoon here. Let me explain this. If you can't read this, it's a group of people sitting around a table, and it says, then we agree 9-1 that we will say our previous vote was unanimous. Now, this is something very careful you have to think about in taxonomy. So I'll use myself as an example. I work in the Rush family. I've written treatments across the United States. So if you looked at Rob Knox's Flora of the Northeast and Alan Weekly's treatments, you could say, oh, they both agree. Well, I wrote those. That's kind of unfair to pad the information like that. So oftentimes we have to be careful to make sure we're using an individual's taxonomic concept, not to be redundant with those taxonomic concepts. So our ITU mirrors a grade school scaling from A to F. So a taxon like A is something like Franklinia. It is universally accepted as a distinct entity. We will not find a treatment that lumps Franklinia. Occasionally, Franklinia is placed in the genus Bordonia, but it is always recognized, Alitamaha is always recognized as a distinct species. Then you have plants like Juncus perfidus. This is a Massachusetts plant. This is very rarely lumped in floras and manuals. Most of the time, more than 50% of the sources will recognize this as a distinct entity. Then you have groups like category C. These are species of minority taxonomic acceptance. And the reason we went with this category as well is that we have entities like Narthiki in Montana. Most treatments do not recognize this as real. But then you have numerous treatments from numerous different authors who disagree. This is where I think science needs to be vetted a little further, because these are areas of debate. And then we have categories D and F. These things, for the purposes of this paper, we didn't look at these. We maintained a list of these because these could be very important endemic and now extinct plants. But some of these plants, like Penstemon leptanthus, which is category F, was described by Fennell, and no one ever recognized it again. It's often not even put in synonymy, so we're not sure if anybody even vetted that name. So that could be a distinct, extinct plant right now, or it could be not meritorious. So those more dubious names are put into category D and F. So for the purposes of this paper, if you break down all these names, we used categories A, B, and C for this paper. If you add those together, you get 64. And then we had another plant that we couldn't calculate an ITU on because it was so recently recognized as distinct. We only had one person's opinion. So we were very clear about that. That gets an NA, not applicable. So we have 65 taxa that are presumed extinct from the United States and Canada. And visually, I find this pretty fascinating because botanists, as you can imagine, don't always agree on what a species is. But when it came to the extinct plants, a lot of them were unanimously recognized as distinct entities. And then we have a whole huge lump. That's not even accurate at this point. I need to update the slide. There were many, many more Ds and Fs that don't pass scientific muster. And then you have this area, I call it where the action is, between B and C. Those are things that the scientific community doesn't quite agree, and there are less of them than I thought there would be. I expected the area of disagreement in B and C to be much bigger than category A. Here are some results. We have 51 species, including 65 taxa, so 14 infraspecies, that are believed to be extinct or globally historic. And that is more than 100%. It's 108% more than any previous study that looked at extinct plants in North America. We have 42 taxa that were global single-site endemics. That is 64%. So I dare say the majority of these plants were only known from a single place on Earth before they went extinct. But that brings some questions of data limitation. Were these really single-site endemics, or were they only collected by one person one time? Only 2 of these plants were listed by the IUCN Red List as extinct. Now, the IUCN is the international categorization of conservation ranks. Now, in North America, we're very lucky we have NatureServe, but it's still surprising to see that the international standard only have 2 of these listed as extinct. So if you look at the entire flora of the US and Canada, which is around 15,882 species, that's a pretty specific number to be an estimate, you'll see that about 0.4% of our flora has gone extinct using our data. But it's hard to come up with an extinction rate because you don't know exactly when things started to go extinct. So for instance, if you look at Franklinia, it was last seen in 1803, but we don't know when it went extinct. We just know it's extinct now. So we have to kind of assume, and if you assume 1804, you're looking at about 216 years, that needs to be updated. That's about 0.3 taxa per year are going extinct, or 3 per decade. So that gives us a pretty nice baseline. Now let's look at some more of these results in detail. Now for the non-botanists out there, this isn't very surprising to see the biggest families lost the most in some cases. So the daisy family had 8, the bean family had 7, the rose family had 7, and the brome family, Boraginaceae, had 6. Now in New England, we don't have any bromes. This is largely a Western genus, which is why this is so large. There are some noticeably huge families at a global and North American level that didn't see many extinction events, like the grasses, 2 grasses, 1 sedge and 1 orchid. Well, I can speculate why the grasses and sedges are so underrepresented, and this is likely an issue with collection effort. Grasses and sedges are largely avoided by botanists. They just don't like them. I love them. I pay great deals of attention to them, so I'm kind of sensitive to this concept. So this is most likely a complete artifact of collection and data limitations. If we look at the genera that have been lost, you'll see the hawthorns had 4. Now, the hawthorns are a very difficult group. There may be more. There's a lot of taxonomic challenges in this group, and it is probably the most poorly understood genus in Eastern North America, in my opinion. So we had 4 Crataegus, 3 Astragalus, and then very interestingly, we have 3 Cryptantha and 3 Plagiarobothra. Well, why is that surprising? Well, these 2 genera, which are among the 4 largest, represent all of the Boraginaceae. This entire family had 2 genera represent all of their extinction events. And even more interestingly, all 6 of these extinction events are known from 1 subtribe in that family. And these two genera are apparently sister genera. So the odds that all 6 extinction events in this family are evolutionary sister genera is astronomical. So I expect something happened here with phylogenetic consequences, maybe a pathogen. I don't know at this time, but this is a really surprising discovery from the study. So let's map out these extinction events and talk about what we can see. So first, I want to point out that there are only 2 plants that had broad geographic distributions. This is Micranthemum micranthomoides, the Nuttall's micranthemum. That started my love affair with extinct plants. And this is Crataegus fecunda, which is extinct in the wild. We'll talk about that a little more, but the only other one that had a broad geographic range, which is defined here as being in 4 or more states. I will point out that this map has a very strong southwestern tilt. This is most likely an artifact of data, in that settlers landed in the east and progressed to the west. Botanical inventory in the west has a much longer history than here in the east. As you can imagine, the first settlers of European origin who landed here settled the open, grassy habitats that would have been easy to till and convert into settle, and that's most likely where your narrow endemics were, or your rare plants were. So the habitats in the east were completely altered before they're ever botanically inventoried. Where in the west, at least we have early studies like from Lewis and Clark, and we also have a great deal of diversity here in the southwest too, so it's probably multifaceted. But I would like to point out the strong southwestern tilt of this map. A little surprising that the breadbasket doesn't really have a lot of extinction events, but frankly, not to cast stones at their flora, it is not as diverse as many other areas in the country. Before, not surprising that the Apalachicola region or even the Lake Wales Sand Ridge, which are famous areas of endemism, do not have extinction events. And then I talk about this when I'm not just speaking to the New England Botanical Club, but it's also fascinating that New England has so many extinction events compared to Florida or other areas of the US. So we'll move on, just quickly look at some of the bigger losers, the biggest losers. And if you had to predict it, you would have guessed California, and you're right. California has a hyper-diverse flora. They have broad coverage from north to south in their state, and they lost 19 taxa. Texas, which you may have also guessed would have been the second largest because they have an enormous flora as well and huge geographic coverage in the country, has 9 extinct plants. But I want to point out these 2 are also largely southern in distribution, which is a little surprising. And I hope, though the data doesn't suggest it, I hope some of these plants are only extirpated from the United States. There's possibilities, And like Quercus tardifolia here, or some of these other ones in Southwest Texas may still be in adjacent Mexico, but the data at this time does not argue that. Now, the third place, we have a tie with New Mexico and Florida. And I want to talk about New Mexico because it presents an interesting issue with some of these extinct plant species. So there are 4, but one of these we really can't even map accurately. The lost sunflower, which is aptly named Helianthus praemetermis, is unmappable. The label says New Mexico, but the collection party was clearly in Arizona on the day it was supposed to have been collected. So we don't even know where this sunflower came from, but taxonomic experts agree it's distinct. We just don't know where it came from. So extinct plants in old collections pose very strange and unique problems sometimes. And then Florida. The fourth, or the tied for third, 4 extinction events, all located in far South Florida, one from the Everglades, Miami, Rockland, the beach type habitats. Florida, as you know, is a very diverse state. But again, the Apalachicola Panhandle region of Florida surprisingly didn't have anything. And then the Lake Wales Seine Ridge, if you don't know about this, this is an ancient archipelago or island chain. And there are very narrow endemic plants distributed up and down this exposed ridge of sand. Again, no extinction events, but this is likely an artifact of data. I am sure more than 4 plants went extinct in Florida, but there were probably dark extinctions or they went extinct before they were ever documented by man. Now we get to New England. As we all know, it's a 6-state region. I grew up right about here in Claremont. I didn't know there were extinct plants in New England growing up. I wasn't the biggest kid into botany, but I was on the Envirothon team, so I kind of like nature and hiking. But I thought, as a professional botanist now looking back, I would have known something about any extinct plants, but it just never came up. So I kind of wonder if some of these plants are under the radar, because over the years I've talked to various botanists in New England about some of these plants, and I'm always surprised when somebody doesn't know one of their extinct plants in their state. And it's also surprising there are 5 extinct plants in New England, only 4 in Florida, because New England isn't really a center of plant endemism. I mean, you get some endemics, but it's not like Florida, which has literally hundreds of endemic plant species. So this is a bit surprising, but I also find it very kind of cool that Fernald, who I have the utmost respect for, described 3 of these plants. So Fernald was the botanist at Harvard. He wrote Gray's Manual of Botany in 1950. He did a lot of work from like 1890 to 1950. Phenomenal, brilliant botanical mind. So it's fascinating to me that 3 of these taxa that still pass scientific muster were described fairly recently by the keen eye of Merritt Fernald. So we're going to work our way north to south, looking at all these extinction events in New England, and coincidentally, these also come up in alphabetical order. So the first one, outside of Burlington, Vermont, is Robbins's milk vetch, Astragalus robbinsi variety robbinsi. This is a bit peculiar that the type variety is extinct, but there are 7 other varieties still extant in North America, and 2 varieties, at least 2 varieties in Vermont. Variety minor is the one I think that you encounter still in that region. So this has an ITU of A. It is universally accepted as a distinct and now extinct entity. This was a global single-site endemic from the Newski River. And if you look at the old herbarium specimens, it says Onion River because that was the original name of the river. But this went extinct when a dam was constructed, flooded the habitat, and now the species is lost forever. Now, I will say I have a side project I call Project Phoenix, and we're trying to take seeds off of old herbarium specimens and try to propagate them. And I'm working with Valerie Pence at the Cincinnati Zoo Botanical Garden on this. And this sheet here is from the Field Museum in Chicago. You'll see there are ample seeds on this. And just recently, that we're trying this one again. Some seeds from this specimen were mailed to Valerie. So hopefully we may be lucky enough to resurrect this extinct plant. We'll keep you up to date on that, of course. Moving to the south now, we go to Manchester, Vermont, where we find the smooth pagoda plant. This is Blophilia hirsuta variety glabrata. Now, this has an ITU of B, so there are some treatments that do not recognize this as distinct. This is starting to trend toward C, because I think Art Gilman's most recent treatment lumps this, the flora of Vermont, but I need to check that. But this is known, another global single-site endemic from Manchester, the dry wooded slopes of Mount Equinox and the dry wooded pastures at the base of Mount Equinox. It is the only place this plant's ever been encountered. There have been some interesting and almost intermediate specimens recently collected by Aaron Marcus, a Vermont botanist, with their heritage program. And he has sent material to myself, Aaron Foden at Missouri Botanical, and Derek Poindexter at University of North Carolina, Chapel Hill, because we have an undescribed Bophelia here in the Smoky Mountains that we'll be describing this year. And we're hoping to get ITS, some molecular data from the specimen to compare it with, because we want an answer to this question. Is this a valid, meritorious entity, or is it just a weird form of Bophelia hirsuta? So this was last seen in 1932. We don't really know why it went extinct, but this is another one of the Fernald descriptions described in 1899, last seen in 1932. So good on Merritt Fernald for describing this, which gives us a hypothesis to test here in the future. And hopefully we'll have some molecular data to vet this plant's taxonomy against. Moving further south, we now go to Manchester, New Hampshire, where we get the smooth, slender crabgrass, Digitaria filiformis variety Libiglumis. Now, for the non-botanists out there, yes, we do get very rare native crabgrasses. These aren't just weeds in your lawn. And this is a great example. This is a single-site global endemic known from the rock rumen in Manchester, from dry, peaty, hollow granitic ledges. The last specimen, clearly 1931, thin humus in soil depressions of granite ledge. This has an ITU of A, universally accepted as distinct. If you look at the Natural Communities of New Hampshire, the Sperduto 2011, he calls this a temperate ridge cliff talus. So a very interesting habitat type, 30 meters, very large. And Bill Nichols, New Hampshire's botanist, Anne Francis from NatureServe and I, who I think are both in attendance, hi guys. We have a manuscript in prep where we're going to talk about this plant because we called it globally historic for our paper on conservation botany, but Bill was able to get back there recently and do some more survey work, and he kind of agrees this thing is unfortunately now extinct. So why did it go extinct? We don't know for sure. Knowing why extinction events happen is very difficult. And I bring this up, and this is a sensitive matter. I'm a huge fan of Fernald. And looking back at what happened in the 1920s as a botanist is very different than what you do today. But Fernald really baled some hay. We have found 28 herbarium sheets that he collected of this plant, all with roots. And there is probably no way that this plant could have supported this level of collection effort. So though it's speculative, it could have been over-collected, and part of the reason why it went extinct. Now, I do have an asterisk here because this plant was recently reported as being rediscovered in Mexico. I have serious doubts about that, at least from an evolutionary standpoint. There's very little likelihood that the plant in New Hampshire and the plant in Mexico are of the same phylogenetic origin. And my colleagues at the Smithsonian, Paul Peterson, Rob Soring, agree with me. Like, they need to really study that stuff in Mexico. So I want to make that known. This is reported. But Bill Nichols sent me some really nice pictures of this rock rumen habitat. These are from 2012. Here it is looking up from the park. And there is Bill Brumbach, one of, I assume, your members, who worked for, I think it's now Native Plant Trust, who was in New England Wildflower Society for a long time. But what I find fascinating about this photo, and I never even thought about this, is if you look at those little rocky pools here, you can kind of see them. Those are Digitaria, those are crabgrasses, but they're non-native species. So it's quite possible that these non-native crabgrasses outcompeted this very rare and local endemic species. But again, very hard to know the cause of an extinction event. Moving further south, you get to the Cape, where we find Juncus purvidus. Now, this has an ITU of B. It's almost universally accepted as a distinct species. It's a single-site global endemic from brackish marshes near Hyannis. As described by Fernald in 1920. Now, very rarely, this plant, like Gleason and Cronquist did this, they assigned this purvidus to the European species Juncus subnodulosus, saying that this is just a waif, which makes sense because Hyannis is a seaport, like people would have come and go and weird things show up at seaport towns. But the Europeans largely disagree. The snow groups did a paper on this, and I think in the 80s, and they said, no, that is not Juncus subnodulosus. And almost every taxonomic treatment after Gleason and Cronquist recognizes this as a distinct entity. I do as well. I've studied the subnodulosus specimens and purvidus, and I think it is distinct. This was described by Fernald, like I mentioned, last collected in 1927. And I hate to bring this up again, but Fernald really baled some hay. He collected 56 herbarium sheets nearly all of these have roots, and this was a rhizomatous, coarsely rhizomatous perennial. So I'm sure this collection level really impacted this plant, because he talked on the label about the plant just being a few rods in size, which is, I can't remember what a rod is. I looked this up previously, but let's say it was 20 by 20 feet. 56 herbarium sheets on something this rare probably couldn't support that level of collection. But, you know, again, back in Fernald's time, people weren't really thinking about that. So next, in a really interesting story, is down here in Groton, Connecticut. This is Graves beach plum. Now, this is a single-site global endemic. This plant is technically extinct in the wild because it is still found in cultivation, thankfully. It was described way back in 1897, and it went extinct sometime around 2000. And this is, without a doubt, the worst story of extinction we have, because the story I have from the people who did survey work for this plant is there was one big colonial shrub at this beach. It was the only shade at this beach. So people who wanted to use the restroom would use this, if you're catching my vibe. And the plant was apparently just nitrified to death, which is probably the worst way for a species to go extinct. Now, there are two varieties recognized. There's a Meridema and Meridema gravesii, and Meridema still occurs in Connecticut. Now, this also brings up the oddity of ITU level C, these really debated taxa. So close to home, Klooster in 2018 published in Redora a paper showing no evidence to support this using microsatellite data. Wonderful paper. But before that, in the American Journal of Botany, Joey Shaw and Randy Small did a chloroplast genome of the plums, and they showed good support for Meridema gravesii versus Meridema. What do you do with two types of molecular data that show differences? I just present this as ITU level C, because Shaw and Small also in 2005 used haplotype data, and they found that gravesii was more closely related to Perinus geniculata than it was to Meridema. So I don't know what to make of this taxonomic debate. I just present this debate exists, and it's trending towards lumping gravesii with Meridema, but there are still some really interesting data, particularly this chloroplast genome, that argue for its recognition. Next, I want to talk briefly about dark extinctions, which are extinctions that happened before a species was described. We know these happened across the world. Documenting them is very difficult, but since 1994, 4 species in the flora of the treatment area had gone extinct before they were described, and we only know them from old herbarium specimens. This photo is Govinia floridana. This was last collected in 1964, probably over-collected to extinction, but it wasn't recognized as a distinct species until the year 2000. Now, that's happened 4 times, like I mentioned, in the flora. So we have Coriospermum pallidum, which is from the Pacific Northwest. We have Marshallia grandiflora, which I'll talk about here in a second, which was described by myself and some co-authors in 2020. And then Potentilla uliginosa, which is a Californian endemic that went nearly 70 years, plus 70 years before it was recognized as distinct. So the story of Marshallia grandiflora is a very interesting one, and especially when you put it through the lens of extinction. So I know this beautiful plant from River Scour in Ohio, Pyle, Pennsylvania. Seeing this plant is like a botanical rite of passage if you're interested in rare plants in the Mid-Atlantic, because it's a G3. It was proposed for federal listings. I think it's a candidate still, and it has this weird bifurcated distribution of Pennsylvania, West Virginia, then Kentucky and Tennessee. That dot in Maryland is wrong, so ignore that. So you have this weird bimodal distribution. And when I moved to North Carolina, I noticed we had 2 historic counties in the mountains. And that really surprised me, because we don't have, looking at the information we had in our database, I realized it wasn't from high-flowing, high-energy streams like the plants in the River Scour in Ohio, Pyle, Pennsylvania are. Like you can kind of see that is a high-energy, white water, you can raft there type of habitat. And we don't really get that in the locations where this plant was reported from Western North Carolina. So I reached out to Derek Poindexter and Alan Weekly at UNC, and what ended up happening is its own paper we published last year called The True Identity of Marshallia grandiflora, an extinct species and description of Marshallia pulchra. That was in Phytotaxa. This is an open access paper you can get on. But what really excited me is I was studying the rare plant list when I moved here. I saw these records and I went on Smithsonian's website and I was blown away at what I saw. So the plants on the left are what I was used to seeing from Ohio, Pennsylvania. It's a rather kind of dainty but showy plant on the left. And then the plants on the right from North Carolina were much more robust, much larger. Flowering heads are enormous. And the habitat was way off. Like some of these say dry soil, some of them say boggy wetlands. That is not the habitat for the plant in Pennsylvania. So Derek Poindexter and Ellie Clee, you see, because they recently described a narrow endemic Marshallia called Marshallia legrandii, and I asked, have you studied the type specimens of Marshallia grandiflora from North Carolina? They hadn't. I sent them this material, and they were equally blown away, so we started to study. We found numerous differences. The plants from North Carolina here is have much bigger cypsali, which are the seeds, longer cypsali scales, prominent ridging compared to what's called grandiflora in Pennsylvania and throughout its range. And then for giggles, we compared it to Marshallia legrandii here on the right, which is a diabase species only known from 2 places in the world, in North Carolina and Virginia. So we did a bunch of morphometric analysis, looking at seed shape and leaf size, etc. And we found 3 nice groupings. We have this North Carolina grandiflora here in X's. We have the typical grandiflora from the Corvus range here, the extant material. And then we have legrandii here, which is the diabase thing that everyone's accepted as different. And when you lay these out on the map, they make perfect sense, because you have Marshallia, what we now call Marshallia pulchra, which is restricted to the Appalachian Plateau physiographic province. You have grandiflora, which was endemic to this 2-county area in the Blue Ridge physiographic province. Then you have legrandii, which has been recently described in very rare plant diabase variants. Now, interestingly, I don't go into it in the talk today. There are 3 extinct plants known from the same small area of Western North Carolina. So this is what we are calling a developing extinction hotspot. I expect more things have gone extinct from this region that have been well documented. Now, just to lay all 3 of these side by side, we have what we now call Marshallia pulchra on the grandiflora in the center and legrandii on the right. And now for you nomenclature taxonomy people, the type specimen, or what described Marshallia grandiflora as a new entity, was from Western North Carolina. So the name grandiflora had to apply to our Western North Carolina material. So we had to rename the other stuff, which we've now called Marshallia pulchra. So dark extinctions are out there. If you see weird distributions on range maps, consider the possibility. That's one thing I've learned about moving to the south. There are undescribed species all around, and you have to be open to the option that something could be new and undocumented. Now, another really exciting component of this work was the discovery of all these plants that are extinct in the wild. We call those EW. That means they're known from the horticultural trade. And for the purposes of this paper, we defined extinct in the wild as something with no naturally occurring populations. And that's an important definition we'll talk about here in a minute. So we had 4 EW plants. We have this EW with an asterisk, which is Crataegus lanuginosa. This is because I put an asterisk on this because I personally have not seen this plant. It comes from good authority. Ron Lance, who's a hawthorn expert in the Southeast, told me he planted this in a garden a handful of years ago. It's actually at 2 gardens. And now this material has been mailed with scion wood to the Morton Arboretum in Chicago, where it's being propagated. So hopefully it will not completely go extinct. We're getting some redundancy there. And then we have these 2 plants that are EW question mark. These are reported from gardens overseas, but they never replied to my emails and inquiries about the identification, because a lot of gardens unfortunately misidentified stuff. There were all sorts of weird things in the literature as being reported as in gardens that are just misidentified, that I won't go into today. But the story of Arctostaphylos franciscana is a fascinating one. This was described by famous California botanist Allison Eastwood in the 1920s, I believe, and then it went extinct and it was lost. And then one of the co-authors on the paper, Dan Glausenkamp, rediscovered this. Very exciting. The problem, they discovered it right on a roadside. And this is called Doyle's Avenue. And this area leads to the Golden Gate Bridge. And this was back in 2007, 2008, when shovel-ready projects were really the buzz because of an economic recession. And there's no way the rediscovery of this plant was going to shut down this shovel-ready project. So the community out in California did the best thing they could, is they dug this plant up and moved it. And it's kind of sad we couldn't protect this plant in its natural habitat. I'm glad it's moved down the road and it's doing well, but this is technically a gardened plant at this point. So it meets our criteria of no naturally occurring populations. I've alluded to this, but knowing the cause of an extinction event is very hard to determine. Even when a plant species was known from a single spot in the world, like these global single-site endemics, unless that site was destroyed, it's really, you're inferring why it went extinct. So even Franklinia altamaha, that famous shrub that everyone knows about, we don't know why it went extinct. We think it might be because of fire. We think it might be from habitat destruction. We think, we don't know. And as scientists, we like to know. So unfortunately, pinning the causes of these extinctions is very difficult, but it's pretty obvious in many cases that human causes are playing a role in these. Now, it's even harder when you have broad-ranging species like Crataegus fecunda and Micranthemum micranthomoides. Micranthemum micranthomoides was in 6 states. We have no idea why this plant disappeared. It wasn't even considered rare until the 1980s, when heritage botanists in all these states are starting to compile rare plant lists, looking at each other, asking, has anybody seen this? That we realized it was already gone. We think this was the proverbial death by 1,000 cuts. You have ditching of wetlands, you have riprapping of shorelines, you have Phragmites taking over habitats, you have potentially non-native algae blooms. All sorts of things are probably caused the extinction of Micranthemum micranthomoides, but again, it's hard to know. So part of this talk, I want to bring up that this can be a call to action. This should be a call to action, because preventing extinction is the lowest bar for success we can set as conservationists. When you think about what we do, we're trying to conserve the natural resource. And extinctions, species falling off, is really unacceptable. And we're just not always being successful in preventing extinction events. So I'm working on a project now to prioritize these single-site global endemics for ex situ, which is outside of the field, and in situ, which is in the field conservation efforts. So why do we need to prioritize? Because we're not doing a great job at that currently. So this plant on the left here is Sarracenia aureophila, beautiful pitcher plant species. That is, we're done with this plant. So this is a globally rare pitcher plant. It's charismatic, I understand that. And if you go to the Botanical Gardens Conservation International database, where you can search any plant and just see how many collections conservation gardens are reporting to have, you'll see we have, for Sarracenia aureophila, there are 44 ex situ sites across the world. That's fantastic. That is actually more than all of the natural populations in the world. So I expect every population has been safeguarded and is backed up. But if you look at a very rare, narrow, single-site endemic, Pachyra serpenticola, which is known from Western North Carolina, there are no collections in all of the BGCI holdings. I think we as heritage botanists can help draw attention to these very narrow endemics that need conservation. Because these single-site endemics, as we saw, are the most likely to go extinct because all your eggs are in one basket. And there's a nice picture of Pachyra serpenticola, which I'll talk about again at the latter end of the show. Collaboration with Anne Francis and Amanda Trayer at NatureServe, we have a methodology developed that I'm not going to go into now, where we're trying to identify all these global single-site endemics of the United States for ex situ and in situ conservation, to make sure those sites are protected and that the seeds are backed up into conservation gardens and seed banks, etc. But we're still vetting this data. We expect to break these data sets up and disseminate them among botanists in different states for confirmation that our methods hold. So if you look at a nice map of NatureServe, they have this map of biodiversity importance, MOBI. This is essentially a hotspot map of imperiled plants. So you can see the whitest areas are places where there's a lot of imperiled plants, and then these cooler areas are places where there aren't imperiled plants. The global single-site endemics correlate very strongly to this. There are 29 for California, which is clearly the high water mark, and then you step down to Texas and Utah, which have 12 and 11, and then a scattering of single-site global endemics. Now, the one from Vermont, that is Isoedes viridi montana, which you may or may not recognize as distinct, which is the green mountain quillwort, which is known from a pond at Haystack Mountain. And again, I don't believe that's in conservation anywhere. So that global single-site endemic probably should be backed up. So if you want to look at these global single-site endemics and compare them to what's being captured already, you'll see we're falling short. So of the 89 global single-site endemics, only 9 are listed as by the federal government as under the Endangered Species Act. And before you have a knee-jerk reaction to how is that possible, So here's an example. Solidago plumosa is a single-site global endemic from North Carolina. This is the Yadkin River goldenrod. This is a beautiful plant that was thought to be extinct before it was rediscovered in the 1990s by Stephen Leonard and Alan Weekly on individual days in the same week. This plant was proposed to be listed, and the U.S. Fish and Wildlife Service decided not to list it as an endangered plant. Now, I'm not going to throw stones at that. I'm just going to say that is their decision. So just because something has extremely narrow geographic range does not mean it will be protected for conservation. If we look at the Center for Plant Conservation's national collection, they've captured 38 of these 89. And then the Botanical Gardens Conservation Database has 55 of these 89. So you can see we're really kind of falling short of in capturing all of these. So hopefully that project will come out sometime next year, maybe, and we can draw attention to these plants that are under sampled and under protected. So I said this talk was going to be two parts. So that was the extinct plants of the United States and Canada part. And I'm just going to slide over to some biodiversity of North Carolina, because I understand, I talked to my mom who lives in Vermont, hi Mom, that you all are still in the midst of winter and I have daffodils flowering here. So I'll whet your appetite for some really exciting biodiversity. So this map here was taken from Jenkins et al. 2015, who did a study on U.S. protected lands mismatch with biodiversity priorities. So when I moved from Maryland, where I had a great career with the Maryland Heritage Program, I moved to Western North Carolina, people were like, why are you leaving? What are you doing? This map kind of explains it. Because if there's no better place to work in the U.S., in my opinion, and this map argues, than right here in Western North Carolina if you want to conserve biodiversity. Yeah, like I felt like I got promoted leagues, so to speak. So it's great to be here in Western North Carolina. There's still so much to learn. And here are some numbers just to kind of wrap your head around if you can. So we have approximately 4,351 vascular plants in North Carolina. That's about 800 more than New England, give or take. We have 21 endemic vascular plants in the state of North Carolina. We have 27 federally listed species of plants. That does not include bryophytes. We have 20 G1 plants. So that is a category of approximately 1 to 5 populations known in the world. Now that number of 20 does include bryophytes because we have bryophyte hotspots and lichen hotspots here in the mountains. We track as a heritage program nearly 700 plant species. So my learning curve is still really steep. I have a lot to learn and it's really exciting. So my collaborator, Alan Weekly, is right in the floor of the southeastern United States. He maintains a well-vetted data set of what we think will be undescribed species in the southeast. And that list has over 100 plants on it. These are 100 plants that still have no names on them from the unglaciated southeast. So there is still so much work to do because you can't really conserve something unless you know it exists. So that's a really important piece. So North Carolina has undescribed plants, and they're almost all going to be a very high conservation value, that G1 to G3 range. Why are we so special? Well, we have the highest mountains east of the Mississippi. Mount Mitchell, 6686. We have 50 peaks over 6,000 feet. And what some of you might not realize in the Northeast, our mountaintops at that height aren't above the tree line. So they're forested and they can really be hotspots of northern species and endemics. One of the groups that's the most diverse in the world is salamanders. We have 65 plus species of salamanders in the state. It's more than any place on the planet. And I say 65 plus because we're still describing the biodiversity of salamanders. So this here on the right is the Hickory Nut Gorge green salamander. This was described just in 2019, so just 2 years ago. This has a very narrow distribution here of this Hickory Nut Gorge habitat in Western North Carolina. There's a little inset for you to give you an idea of where we are. But that isn't even the most recently described salamander. The most recently described salamander in North Carolina is the Sandhills two-line. This guy's a little beauty, isn't he? This is found in the Sandhills region, where in the Sandhills is where the coastal plain physiographic province meets the Piedmont physiographic province. You get the fall line, or more appropriately, the fall zone. And in this area of the state, we get the Sandhills region. And these little red dots here, which are located all here, are this new species, the Sandhills two-lined salamander. That was described just recently in 2020. And I have on good authority from some people I know who work with salamanders, there are at least 2 more entities that need to be described in North Carolina. We have a matrix of high quality forests that still exist in Western North Carolina. We have more old growth forests in the mountains here than any other state in the Southern Appalachian. I mean, the Great Smoky Mountains themselves have more than 1,400 plants and more than 100 species of tree. And I understand why the Europeans are so overwhelmed when they first explored these habitats, because the Great Smoky Mountains have more species of woody plant in the Smokies than occur in all of Europe. So I made the joke before this conference call started that glaciers, thank God our mountains are oriented north-south largely. So as the glaciers move south, the diversity could move. In Europe, their mountains are located east-west. So as the glaciers moved, you had huge extinction events. And that's one of the reasons our diversity is very high compared to Europe. Now, looking across the state, we have an amazing coastline. We have the second longest coastline next to Florida in the east, including this beautiful little guy, Amaranthus pumilus, which is known historically from Connecticut and Rhode Island, I believe, but we still have extant populations here. This is federally listed. And probably the most charismatic, exciting plant everybody knows, Venus flytrap, is a naturally occurring species here in North Carolina. I'm always surprised when people think this is a plant of the tropics, but this is one of those plants that was really transformative for me to see in the field as an undergrad. I remember going to the Green Swamp in coastal North Carolina and just seeing Venus flytraps in the wild, and it was really cool. But two of my favorite quotes, like Charles Darwin called this one of the most wonderful plants on earth. And then Ellis, in this beautiful letter to Carl Linnaeus, said, "Miraculum natura, a miracle of nature." It really is an amazing plant. Everyone, I hope you all get to see in the field someday. So let's come back up to the mountains. This is where I work, and it is an amazing landscape. This is the Linville Gorge. This is a really underrated area. My first trip to Linville Gorge was just this last year. So you have this gorge, and on top of the gorge, you have this really dry pine oak heath type habitat, really pyrophytic, fire-dominated landscape. And to come down the gorge, you get more music. This location is the only place in the world for the mountain golden heather, Hudsonia montana. In New England, you get two species, Hudsonia tomentosa and ericoides. In my opinion, I've seen them all. This one's the most striking, really big flowers. Inland, very strange, but endemic to just this one place in the world. I'm only going to go through a few more sites, but every species I'm going to talk about here is federally listed. And I went with Grandfather Mountain because it tells a good story about all the crazy stuff we get. And it's kind of similar to the man on the mountain, like in my New Hampshire roots. You can kind of see that face. You have the chin and the nose and the furrow, the brow. And that's what they call Grandfather Mountain. So this is pretty high, 5,500 feet up in the northern part of the mountains here. A huge area of protected land. We're talking that's closer to 30,000 acres now of protected land at the state park and TNC and the U.S. Fish and Wildlife Service all work together to protect this amazing place. So what's found here that's so amazing? Well, we have Virginia big-eared bats, which are federally listed. These are seriously in decline because of white nose, and there's a range map for you. And then one of the craziest organisms I've had the opportunity to work with is the spruce fir moss spider. This is a federally listed species. That is the size of a BB. It is about 0.1 to 0.15 inches. Like, it is a tiny tarantula. Like, I wouldn't have known that was a tarantula, but it is. And when I say spruce fir moss spider, you might say, really? Spruce fir in North Carolina? We do at high elevations to get spruce, red spruce. We also get fir, but not where you're used to. We get a very interesting and southern endemic called Fraser fir, Abies fraseri, which occurs at the mountains from Virginia, Tennessee, North Carolina, and very rarely in Georgia. Now, there are species here at Grandfather Mountain which you might recognize, the genera, GM radiatum. This is almost endemic to North Carolina, a couple sites in Tennessee. We call these high elevation rocky summits. They can be very treacherous to survey. I think that's Gary Kaufman, my colleague from the Forest Service, on just a vertical cliff inventorying some of these very rare plants. This is another federally listed species. This is sister to GM peckii, which you all get in the White Mountains here on the right, and it's also in Nova Scotia. And then it looks very similar and sister to GM calcifolium, which is in Alaska, Canada, and then in Japan. This photo is actually from Japan, in a very similar habitat type. And all these plants I'm going to talk about kind of co-occur, like real hot spots for all these really narrow endemics, including the mountain bluet or Rome mountain bluet, Houstonia montana. This is a real pretty plant. Houstonia purpurea is a broad-ranging species in the southeast. And this is either sometimes called subspecies montana or recognized at the species level. It's Houstonia montana, as I prefer it. Is a very narrow endemic to these same habitats, North Carolina, Virginia, and Tennessee. Co-occurring with this is the absolutely stunning Heller's blazing star, Liaturus helleri. And co-occurring with that is another federally listed plant, the blue ridge goldenrod, Solidago spicamia. So these are all extremely narrow species of a narrow habitat type in a narrow geographic range. And let's not forget, we get the only federally listed lichen, the rock gnome lichen. This is called Gymnoderma lineare. It has a new scientific name, but we don't use it because we like to match the Endangered Species Act. So not only do we have well-documented, well-surveyed, incredibly important biodiversity areas, we have areas that we're still discovering how important they are. And this is the Buck Creek Serpentine Barrens. This is one of my favorite places on earth. You might not know that we have serpentine in North Carolina. We do. Serpentine is a bedrock geological formation, an ancient base in the ocean that's been produced through uplift or exposed through uplift, ultramafic, heavy in the metals, heavily pyrophytic. And we get really strange disjuncts. So things that you all would think were common, like Deschampsia cespitosa, you'll see are remarkably disjunct way down here to the Serpentine Barren in western North Carolina. So this is very common in the west, in the northeast, and then jumps way on down here to western North Carolina. Same with Elymus trachycollis, another very strange disjunct, which is very common for y'all. And not to ignore the pretty flowering plants, fringe gentian, very beautiful, very rare for us, more common for y'all. But we also get Midwestern disjuncts like Spirabolus heterolepis, the prairie dropseed. You see it's kind of scattered in the east, including Clay County here, and more common in the prairies, hence its common name. We get stunningly beautiful plants like large-leaved Grassa parnassus, which is just pretty uncommon everywhere. It's not tracked in Missouri. This may represent more than one species in this geographic range shown up there about the Bonac map. But this is still one of my favorite plants because it's just so beautiful. But we're still pulling narrow endemics from this site. This is Ryan's aster, Symphyochrome ryan, described by Alan Weekly and Tom Govis. I think it was back in 2004. The story goes they were surveying this habitat. His daughter Ray was a little colicky, so they had to stop in the field to tend to her. And I think it was Tom said, "What is this aster?" So they kind of named it playfully in her honor. This is a global single-site endemic only known from the Buck Creek Serpentine Barrens. Packera serpenticola, which I mentioned earlier, described by Harvard botanist Dave Buford et al., described Packera serpenticola, the Buck Creek packera, the Buck Creek ragwort, another global single-site endemic to the Buck Creek Serpentine Barrens. This plant forms a carpet of vegetation in places you cannot help but walk on it. So the endemic plants of this habitat are very common. There was even a paper in Systematic Botany probably 10 or 15 years ago that talked about how narrow endemics can be common where you find them. This is a great example of that. But we're still describing plants. This is an undescribed species of heartleaf, hexastylus, or acerum, depending on how you want to treat this. It is a clonal patch-forming plant, thousands of individuals at the site. The leaves are not mottled the way every other hexastylus is. We actually have a bunch of specimens collected for morphometric analysis now. Our molecular data is solid, and there are obvious characters. So this plant on the left is the Buck Creek heartleaf. You'll see it has, these are the stamens here at the base. These are the styles, and they have long stylary appendages. Whereas Ruthii here on the left and Aerifolium, which this plant has been confused with, do not have those characters. This is easily distinct in an undescribed single-site endemic of western North Carolina. We hope to have that published this year. And there are 2 more plants under study right now. When I went out to the Buck Creek Serpentine Barrens for the first time, I was blown away with this blue-eyed grass here. This is not a grass at all. This is a member of the iris family Cicericium species Nov. We are pretty certain this is an undescribed species, but the jury is still out. We need a little bit more data. There's also a thalictrum, a meadow rue, which is very common here at the Buck Creek Serpentine Barrens. That is most likely undescribed. Undescribed species are all around us still in the southeast and probably the northeast. We just have to be willing to have the opportunity and to think about things as potentially undescribed, not shoehorn them into other areas. So in conclusion, there is just so much left to do. I feel kind of bad about giving short shrift to the North Carolina biodiversity, but I didn't want to keep you here all night because that could be its own distinct talk. But so there's still so much more to do. Extinction is more common than previously documented, clearly show that. We have these 65 taxa. Our global single-site endemics need to be prioritized for conservation. Now, I know there are a couple of critiques we're still vetting from New England that may meet that list of global single-site endemics for conservation. But keep thinking about what might be of really high conservation value in New England, because we need to think about in-site conservation, in situ conservation, and outside the site or ex situ conservation, because they're both critical components for preventing extinction. Because I'm really afraid taxa are slipping through the cracks. Since we published this paper, we have 2 other plant species that might already be extinct that no one kind of knew about. So this work is not done at all. We all need to work more collaboratively because the stakes are just way, way, way too high. Acknowledgements, all my co-authors, of course, but the Smithsonian was great to let me just camp out and go through literature. Maryland Heritage Program, my previous career, and North Carolina Natural Heritage Program were great in supporting this work, even limitedly. Gary Moore at the USDA Plants Database, one of my co-authors, found the money to make that paper an open access paper, so he gets a great deal of credit. Ron Lance is a noted Crataegus expert. He really held my hand through that genus because it is the worst. Abby Myers and Nick Barton at the BGCI helped a lot with data vetting through their conservation database. Matt Lobdell has really been helpful with trying to propagate some of these Crataegus that are now extinct. And then if you want to follow up more about this work, Marion Manal did a little piece in the New York Times you might have seen on extinct plants, and Susan Millis did a Science News article that was great. And then I was on the In Defense of Plants podcast. So I really need to thank Matt Kandidis also for plugging the story and draw attention to it.
Thank you so much. That was just a fascinating talk. The tour through North Carolina really capped it off. Great. There was one comment that came in to me directly earlier in the talk about whether there are issues with any of these species of gene flow from related species in horticultural settings. So, narrow endemics that might have close relatives that are in cultivation or horticulture. And were there any in this set of species you saw where that was a concern of this hybridization issue?
Not that we know of. That could be a problem with Crataegus, because Sargent, who described literally hundreds of Crataegus species, he did a great job in distributing living material. So Crataegus fecunda was something he sent to numerous herbaria or arboretum, and that's why it's extinct in the wild. That's why we have this weird record of Crataegus delawarensis in Europe that we can't dismiss because Sargent mailed material overseas. Like there was something at the Jardine Gardens. I think they have a duplicate of Crataegus fecunda living. So it gets complicated with plants like that easily hybridize because we don't know what they really were in the first place. And now you have this living tree. So we don't know if it's a one-to-one. And I even get further confused because you're grafting one organism onto another. Like, I don't know what the implications are in any of that. I just have to kind of listen to the Crataegus experts because you're getting way above my pay grade. But as far as we know, that isn't a problem. But I am fascinated by the phylogenetic implications of extinction. And like Brent Mischler et al., they did a phylogenetic analysis of North American plants, and they're going to try to expand that project. And I'm going to be involved with them doing, hopefully looking at what we've lost from a phylogenetic standpoint. Because things like Thisnia, which I didn't even have time to talk about, that must be incredibly long branched, because that is a family we don't even get other than this one extinction event. So not that we know of is the short answer to that question, but it's certainly possible with things like Crataegus.
So there's a question here from Asher. So basically asking a really fascinating question about how important or possible might be reintroduction or renaturalization of some of these species that did survive in horticulture. So something like Franklinia or the Arctostaphylos in California, are there discussions of reintroducing these to sites where they existed formerly or establishing populations in the wild?
I believe that's been tried in Georgia with the Franklinia, but it hasn't been successful. So it kind of lends itself to something that's happening here that isn't just as simple as this plant went extinct because of some simple cause. Now, I'm not an advocate for planting stuff around. I think anytime we plant something, it needs to be very clearly thought out, and the whole community should kind of vet that idea. We should document it. It should all be above board. I think it's a topic that we're going to have to get more comfortable with, because one of my side projects is to look at all of the potential extinction events in Florida, just looking at sea level rise predictions, because they go from 20 to like 30, they go from 4 to like 30 in the next 20 to 50 years, just looking at the conservative projections of sea level rise. So we either need to stand by and let it all go extinct, which I don't think we want to do, or we have to come up with a plan to propagate these things or translocate them to suitable habitats inland. So I don't cast dispersions on that idea, but we just need to be really careful anytime we want to move stuff around like that.
Yeah.
But it's great that we have the opportunity, because with most of these plants, we can't even have this conversation.
Yeah. So there was a question earlier that I had missed, but Matthew asked about your estimate of the extinction rate in North America of 3 taxa per decade over the past 200 years or so. Are there any points of comparison with other regions in the world? Is that high or low, or where does that stack up?
It's high, because the only other paper that looks at extinction rates at a global level was produced in 2019 by Humphreys et al. And they looked at global extinction rates, and my team and I actually did a rebuttal paper to theirs because they really missed some North American data. It just wasn't the best data set. But if you take that paper as gospel, let's say, like you believe everything in that paper, Hawaii is a clear number one, because as you'd expect, so like 95% of their flora is endemic. So many things have been already lost. And then the number really drops. And I think the United States is number 2 now on that list. So we are clearly outliers compared to the rest of the world, but that's a 2-pronged reason, in that we have the resources to inventory what we have. Whereas a lot of countries that are seeing development, and especially biodiversity hotspots, they don't have those opportunities. So it's like a, I hate to say that it's clearly the worst in the United States as a brand spectrum, because we are a rich country that has a lot of opportunity to do our inventory and have academics. So I think we just need to take all that with a big grain of salt.
Great. Yeah. So another question that came in is interesting, and you had sort of pointed this out, but can you talk a bit more about sort of how we might go about thinking about some of these ecosystems that were heavily impacted, transformed before there was opportunity for intensive botanical inventory? So the tall grass prairies in that sort of cold spot on your map of no documented extinctions, and yet you have a region that was totally transformed by agriculture. Any thoughts on that or speculation?
So I don't say this lightly, because I don't like how it makes me look, but we have just transformed everything around us to a point where it's not, I don't think it would even be recognizable. Like, we all have that talk, if I could go back in time and see this place, I wouldn't want to, because we have changed things so drastically. Like thinking about New England, like Fenway Park is built on fens that they filled. Who knows what was there before it was ever surveyed? So I think you get into a weird world of speculation, especially in the South. Like my friend Dwayne Estes, who's founded the Southeastern Grasslands Initiative, like he talks about these vast prairie grasslands, and there's just none of it left. You're looking at roadsides and power line cuts. So I don't know of a way to even speculate about the totality of what is lost. Maybe someone who's really good at theoretical math could do something, looking at what we know of the evolutionary history of all these organisms and modeling, like over time. But I think you're getting to a strange spot of speculation that I don't feel comfortable venturing into.
Yeah. So another question came in. You had mentioned in passing some of these projects that are starting to look for seed material in herbarium specimens, and Asher is wondering if there's any success stories yet from that approach of resurrecting species from old collections.
If there was a success story, you would have heard about it today. It would probably be huge news. But what we're finding, and this is just how herbarium material is treated. It's not meant to be alive. And embryos are living, and they're very sensitive living entities. So we picked that as we've tried 3 or 4 plants, none with success yet, but we're hopeful. So there was a study where someone took some seeds off of an astragalus species. I can't even remember. There were 34-year-old, 24-year-old seeds, threw them in the soil and they grew. So Valerie Pence is a renowned plant propagator. So she's gotten common progenitors and looked at where the embryo placement is in the seed, so she can extract the embryo and plate it and figure out what medium a seed might germinate best on. But right now, we just haven't had success because, I was at the Academy of Natural Sciences, and Ernie Schuyler is there, and he was on my grad committee back 15 years ago. And he's an older guy now, and he said, "Oh yeah, this is a great idea, but our specimens are treated with mercurial, whatever." And it's like, oh, there's no way an embryo survived treatments with mercury. So the odds are really stacked against this idea, but I would love to have a proof of concept because it's rather inexpensive. Like Valerie's estimate on the potential for the resurrection is about $500. I mean, that is cheap conservation dollars that could have massive, massive gain. But unfortunately, we just haven't had luck yet, but we're going to keep trying.
Yeah. Yeah. So, Bob actually just posted a comment also saying that, it's possible your paper and identifying some of these cold spots in the Midwest and the prairies will trigger more research going back into herbarium specimens to try to, identify species that might have been lost. And that's a good point.
And it's depressing to think like that because these dark extinctions must have been more common. And the fact we have 4 of them in the last 20 years, so there's going to be more. We just have to be open to that idea and pursue that idea if you see really weird stuff. I kind of wonder if they're in the index folders, like at the very end, there's just these unidentified things. I wouldn't be surprised if some of those things end up being like, I can't put a name on that. Nobody can put a name on it because it's not described. Yeah.
Yeah. Great. Other questions from the audience here? We've got. Someone did comment that we had Mason Heberling here last month talking about the value of herbarium collections, and that talk talking about herbarium specimens is on YouTube now on NEBC's YouTube. I'm curious to know, you had mentioned sort of this initiative as you're identifying some of these single site endemics and prioritizing some of these species for ex situ collection or ex situ conservation. What sort of planning would go into that with some of those collections? Is the idea to sort of replicate populations in botanic gardens or replicate populations into nearby sites? Sort of what would be the nature of the ex situ conservation you or others have been talking about?
There's really 2 types of global single-site endemic. You have these species that evolved in one place and are only known from one place and will only be known from one place. And those shouldn't be moved around because they're from that place and that's where they belong. But then you have this whole other suite of things that were, and the collection data will show this, they were more common and now we just have one place for them. Mm-hmm. So those are the kind of things that could be translocated back into their old habitats. But the first thing we need to do is identify what these plants are. And I envision, seed bank collecting of these very rare plants so we have some backup should something happen to the only place in the world they occur. Because I don't want to go out and create a bunch of things that are like Franklinia. It's still wonderful we have Franklinia. And no one's upset we have Franklinia. So given just the development pressure and how we're expanding our population and how climate change, you add all these things together, we're going to lose more stuff. So we need to do what we can to prevent the extinction of species. So I kind of envision splitting this list up by state, having the state botanists and the heritage programs kind of vet it to make sure it passes muster, and then kind of working more closely with that conservation garden community. Like here in North Carolina, we have the North Carolina Botanical Gardens and Atlanta, the ABG, and you guys have the New England, it used to be the Wildflowers Native Plant Trust, I think is the new name. Like those groups do fantastic work. And sometimes it's worthwhile to say you might want to work on these things because other groups like the U.S. Botanical Gardens might fund projects that work on this very rare suite of species that are more likely to go extinct.
Mhm.
And then we can start growing them, figure out how they grow, because each one is a different challenge. But then we need to be careful about what we do with the material, because I'm not interested in selling it to people to put in their gardens, because that doesn't really solve, that's not really what I'm into. That's not what it's not a conservation gain to do that. So then you can have the conversation of what do we do with this material? And at a minimum, it can be a living backup,
Mhm.
which I think is really worthwhile to do.
Yeah. So there was a question here from Matthew about, I believe there was a figure you had, and I'm not sure if the number comes from this, but um, approximately what % of the endangered taxa in the US are backed up in seed banks, the Millennium Seed Bank he mentions. What is that number of species that are captured ex situ now amongst the ones you're thinking about?
So that number,
the ones that are in peril, maybe.
Yeah, so we had 89 global single-site endemics, and 55 we believe are in ex situ conservation collections. That's what that BGCI, Botanical Gardens Conservation International, kind of includes all those things. But I'm going to go ahead and put an asterisk next to that 55, because in working on trying to figure out what plants are extinct in the wild, I found some egregious misidentifications.
Mm-hmm.
So every one of those 55 needs to be vetted for its accuracy. Now, I don't like doing this, so I'm going to leave the institutions out. And some of these institutions are massive institutions. Like they reported micranthemum, micranthomoides are having. I'm like, wow, it's amazing. Yeah, we collected it in China in 2007. And I'm like, that genus doesn't occur in China.
Mm-hmm.
There was a specimen, Bochara fruticosa, which is a single-site endemic in Wyoming. An organization claimed to have it collected in Russia. Like the mistakes were so bad that we just need to be really careful with the data. And they were happy to correct their data when I kind of pushed back gently with, hey, this doesn't really make sense. So I kind of wonder if that 55 is correct or not. So that's going to take a little bit of time to vet as well. Looking at that, if it's correct, more than half are already captured, which means we have another half to work on.
Yeah, great. Yeah. Other questions from the audience?
Whoever just said need to go, hey, around until 8:30. That's great.
Yeah.
On a Saturday.
Yes. Well, thank you again, Wes. This is a fascinating talk. We've been looking forward to this for ever since you said you'd come in and talk to us. And it's such an important paper that you put together, and it's going to be an important baseline for years to come. So it's great to see it presented in person and talk through some of these details and have a chance for folks to ask questions too.
Thank you. Thanks for having me.
Yeah, thank you.